When DNA letters get married, who pays the price of the wedding? Turns out it's the incoming nucleotide bride. So, what do I mean? So, basically, nucleotides or deoxynucleotides are DNA letters, and they can join together in different ways. And so, typically, how we think about DNA letters joining are when we're like copying DNA and stuff. And so, you have a template where one strand is acting as a template, and then incoming nucleotides, I'm just going to call them nucleotides, technically they're deoxynucleotides, but they come in, and then this enzyme, so this reaction helper protein, it's going to help bring them together and get them to react in a way that they stick permanently together, and then you build on and you get this growing DNA chain that's like template directed. Sometimes, however, you have two strands of DNA and you want to join them. In this case, you use a different enzyme. You use a DNA ligase. So before you were using a polymerase, and now you're using a ligase. And the difference has to do with where the energy is going to come from. Because DNA letters, so when they're coming in to be added to the chain, they have like money with them. They have a triphosphate group. So you might have heard of ATP as being like the energy currency. But the DNA letters are DNTP, so they have the triphosphate group too, and that's where all the energy is coming from. Because ATP, it has these three phosphate groups, or any NTP has this three phosphate groups, and they're these negatively charged things, and they're like clamped together, like a clamped together spring. And so they don't like these negative charges are trying to repel each other, but they're being held together by these bonds. If you can break them, boom, you get energy, and they can use that energy to pay for things like putting together the nucleotides, which they don't really want to do because you're tying them down, and things don't like to be tied down. So you're like, okay, well, I'll pay for it by giving you ATP or NTP. See, it's really hard to say, like not to say ATP, but to give you energy. And actually, you'll see that the ligase, you actually do use ATP. To help activate, because so when the DNA letters are coming in to be added into a growing chain, they have that those triphosphate groups with them. But if you have two separate strands, and like one of them has and it comes from like a broken strand, one of them is going to have like just a single monophosphate group, because when they join together, they ditch two of the phosphates, so you're left with a single phosphate in between them. So now if you break that strand, now one of the strands has just like a monophosphate, and that's not enough to pay the cost, because now you don't have anything to kick off. And so you have DNA ligase comes in, it's going to add phosphate groups to that monophosphate, boost it up to have higher energy, have something it can kick out, and then it'll join them together, kick out that other thing, and then that other thing will, and you get energy. So yeah, so that was a basic overview. And by things going, I mean like, so when you break off like a triphosphate, you can get a pyrophosphate, which is like two phosphate groups clipped together, and then those can break up to give you orthophosphate, where there's like separate phosphate groups, and that can really help pay the cost and drive this reaction in the forward direction and keep things from going backwards, because now you're like just spent your money, right? You can't spend your money back. And so today I wanted to tell you more about how this is all working at the mechanistic level, and in terms other than poof. Okay, so the two main players we're going to talk about today are DNA polymerase and DNA ligase. And these are both enzymes. So enzymes are typically proteins, sometimes RNA, sometimes a mixture of proteins and RNA, and they speed up reactions without being used up in the process. And so often how they do this is like by holding things together so that they can react, providing the optimal site and that sort of thing. So we'll look at how these enzymes are going to do. They're helping out, as well as what's happening at the molecular level, because although an enzyme is kind of like a lead a horse to water thing, so an enzyme can lead the nucleotides to join together, but it can't actually make them do it, and what they're actually going to be doing the work themselves. But anyway, so with the DNA polymerase, basically what you have is you have free nucleotides. So you have these free DNA letters, and they're going to be joined to a growing chain. With a DNA ligase, what you have is you have like a broken or gap chain where one end has this free monophosphate group. And so it has to be activated with the help of ATP. And then this is going to be joined together. And the reason why we'll see the reason why you need this ATP is because here you have these three phosphate groups already. And so you have this pyrophosphate you can kick off and pay the cost. Here you only have a monophosphate, and when you join these together, you're going to get a And when you join these together, you need something, you're not going to have anything to kick out unless you first activate this chain with the ATP. And sorry, this should have another negative charge there. So this is why you need different enzymes to do these different things, and you need different pathways. Basically, DNA letters, so they're typically just called nucleotides, although technically they're deoxynucleotides because, so RNA and DNA are like super duper similar. Their sugar is slightly different. So basically they have this sugar, this nitrogenous base, and this phosphate groups. And so with ribonucleotides, which is what RNA is made out of, you have this extra sugar, this extra OH group in the sugar. With DNA, you don't have that. And so this has some consequences, like it helps make RNA less stable and that sort of thing. With DNA, you don't have that OH. The other difference is that DNA has the letter T and the base T instead, and the RNA has the base U, but they both compare with A. And the reason why they have the specific base pairing is so what I have is just these like teal squares. These stand in for these nitrogenous bases, and they offer these pairing opportunities so you can have this specific bonding between various pairs of letters. And this is where you get your specificity. But where you get the energy for joining is from these phosphate groups. So when you join together these letters, they link up to form these long chains. And when they form these chains, they're connected by these 5', they're connected 5' to 3'. So we call like the end with this, so we call this position like the 5' position, and this is the 3' position. And it's just based on like the numbering around of the like what's sticking off where on the sugar. And so you can see that the 5' end is going to have this free phosphates, and the 3' end is going to have this OH. And when they link together, they're going to just be linked together with one phosphate group in between them. So this is a phosphodiester bond. And so an ester is where you have like a carbonyl, so a C double bond O next to an oxygen and next to a carbon. Here what we have is a phosphodiester, because instead of having that oxygen next to the C double bonded, instead of having like a C double bond O, we have a phosphorus double bond O, so this is a phosphodiester bond. So these join together, and the important thing here is that when they join together, now they only have a single phosphate in between them. And the reason why they only have a single phosphate in between them is because they use one of those phosphates to pay for the price of joining them together. And so we've talked before about how ATP acts as like an universal energy storage molecule. And the reason why it can do this is because it has these phosphate groups, and these phosphate groups are negatively charged, and like charges repel each other. And so you have these negative charges and they're scrunched together. It's kind of like a clamped spring. So just like if you release a clamped spring, it'll like shoot out. So like say if you had like a clamped spring and you had a ball and you let it go, and the ball would like fly across the room. So you're releasing energy when you unclamp the spring. Similarly, if you hydrolyze or like cut apart the phosphate, then the energy is released, and you can couple this energy release to non-favorable processes like the joining of DNA letters. And so the joining of DNA letters isn't favorable like entropically, because you're taking, so entropy is like a meaning of like freedom or disorder or randomness. And so basically, if you have you have like a DNA strand and you have a DNA letter, and this is all happy flying around, and now you're going to stick it to something. And so you're going to have to pay the cost of removing this guy's freedom. And so you're going to pay the cost by freeing some ATP or some phosphate groups. And so basically, you can use the energy from freeing up the phosphate groups in order to pay the cost of joining together those nucleotide letters. And this is what I mean by like paying the cost of polymerization, because so with ATP, so ATP is one of the RNA letters. So in terms of like those bases, there's 4 DNA letters, A, T, G, and Z. And for RNA letters, A, U, G, and Z. And the A, so the RNA version of the A is ATP, and so the DNA version is DATP. But ATP is kind of like the universal one, and like because all these enzymes have evolved to use it. And but really the thing that matters is that triphosphate part. So although we talk a lot about how ATP does this, you can think of the same sort of thing happening when you have any trinucleotide phosphate, because it's that nucleotide phosphate part, or nucleotide triphosphate, because it's that triphosphate part that's important for doing the paying. And so when you have DNA letters coming together, you have that release of this, pyrophosphate, so these two P's, and then that can then get hydrolyzed to form these orthophosphates. So this gets broken up even further into this orthophosphate. And this is going to give you like a major entropic benefit. So basically lots and lots of freedom, because you're taking these molecules that were clamped together and now you're freeing them, and molecules like to be free. So you're making things happy, and paying the cost. And you're also making it harder to go in the reverse, because so enzymes can catalyze reactions in both directions. Because basically what they're doing is, so you can think of reactions as having, like, you have your reactants, then you have your products. And in order to get from the reactants to the products, you usually have to go through like some sort of uncomfy transition state. And in order to get there, you have to add energy to get over this activation barrier. And so enzymes can kind of lower the barrier to getting there, often by providing like a somewhat different route, stabilizing uncomfy transition states and that sort of thing. But because they're basically just lowering the barrier, they're speeding up the reaction equally in both directions. And so if the reaction is unfavorable, it's not going to happen, or so it'll keep coming backwards. I mean, you want it to come backwards, but you can't come backward. But helping like release the pyrophosphate is going to help ensure that it's not going to come backwards, and it's also going to pay the cost. So this is why this is how you pay for polymerization, is that you have the incoming phosphate group is coming in the triphosphate form. And so you have this guy coming in with his money. If you're wondering about like specificity and stuff, so basically the polymerase, I'll tell you more about like the active site in a minute, so like where I think the reaction is actually happening. But the polymerase has ways to tell if a correct nucleotide is being added. And so by correct, what I mean is that it has this complementarity, that it's the correct. So if it's an A in the template, you want to make sure you add a T. If it's a G in the template, you want to make sure you add a C. And so basically, there's some different ways, and there's a little bit of like dispute about the various specifics, but you have your template letter. So you have your template strand and the letter sticking in. Incoming nucleotides can just like come in, and if they match, they will stick long enough to then have like this reaction happen. And if they don't match, they'll just fly away. But it's not that simple. And it's actually like, if they match, it can make the enzyme kind of position itself so that it is primed for like catalyzing the reaction, so that everything is like in the right position. And if it's not right, then you don't have that, everything isn't positioned correctly. And sometimes it makes a mistake and adds the wrong letter, but then you get like this awkward stuff, because DNA letters, so when they pair in this like specific way. So you have like one of these like these like purines, so the ones with the 2 rings, and one of the pyrimidines, so the one with the 1 ring. And this gives you like this even spacing. If you have like a mismatch, then you're going to have like an awkward spacing. Even if you have even if you have a purine and a pyrimidine, but it's the wrong one, because the bonding opportunities are going to be kind of like misaligned and stuff. So they'll have to kind of be funky, move funky in order to kind of stick. And so you get these bulges in the DNA, and then the polymerase can sense that, kind of shift things into so that the wrong pair gets added into this like exonuclease pocket, and so it can like actually like chew it back and then try again. So there are different ways where you get that specificity, but where you get the actual reaction happening, this is going to happen with the help of some magnesium ions. And so enzymes are really awesome, but they often need help. And so sometimes enzymes use cofactors, which are like helper molecules. And DNA polymerase uses, we call them metalloenzyme. So it uses a metal ion to help it, and it uses magnesium. And so why metal ions are so great? So an ion is a charged thing. And so magnesium is positively charged. And basically they have these big fuzzy electron clouds, and they can give and take electrons, and they can help stabilize like opposite charge things. So if you just need to worry about this magnesium being like positively charged for them. And so your ATP is going to be negatively charged, or whatever your TP is going to be negatively charged. So the magnesium can help stabilize the negative charge. So even though we often think about like ATP is just like floating around in cells, it's actually usually like attached to like magnesium and stuff. So the magnesium is helping to stabilize those negative charges and stuff, because you don't want to like split it apart before you're ready, right? And it helps it from like sticking to stuff and all sorts of stuff. So the magnesium is really important. And so how the magnesium is actually like helped held in the active site by some important acidic residues. And so we talked yesterday about like acidic versus basic residues, and how when we're talking about acidic residues, we are talking about glutamate or aspartate, which are negatively charged. And so you can think about these amino acids as kind of like sticking into that active site, sticking off of the protein into that active site, and they can help hold the metal in place. And then the metal so one of the great things about metals is they can form these like, they can bond to, they have these big fuzzy electron clouds, and so they can you can have multiple things bind to them and like kind of clamp them down into position. And so this is happening in the active site where you have these two magnesium ions. And so one of these is going to help activate one of the end of the 3' end of the growing strand. And then the other magnesium is kind of just going to help stabilize everything as you're getting this uncomfy state. And so how this reaction actually happens is it's an SN2 reaction, so a substitution nucleophilic bimolecular. So I've talked about substitution reactions in other posts, but basically you have, we call a nucleophile is going to attack, and then you have a leaving group leave. And so what do I mean by nucleophile and electrophile? So let me back up a second. And so much more in previous posts, but atoms, so like individual carbons, hydrogen, nitrogen, all that stuff, they are made up of smaller subatomic particles. And so you have protons, which are positively charged, and they hang out in this dense central core along with neutrons, which are neutral. And then around them, you have this big fuzzy cloud of electrons, which are negatively charged. So the protons are kind of like dog walkers. And so they're in the central nucleus, and they're this positively charged thing that are trying to rein in these negatively charged electrons. And the electrons want to be free, and they're all whizzing around. And they're kind of like really energetic dogs, especially the farthest outermost electrons, which we call valence electrons. So those guys are really like trying to break free. So your dog walkers, the protons. So sometimes they're really good at keeping a pull on their electrons. So some atoms are really good at this, and oxygen is an example. So oxygen is what we call electronegative. So it pulls really tightly on those electrons. Other atoms aren't so good, and so the electrons are freer to like go form bonds with other things. And
maybe even leave. And so, but oxygen's really electronegative, so it's going to have a tight pull over electrons. When atoms can join together to form covalent bonds, so those are strong bonds, like the types of bonds that we're forming in these reactions. So they're not just like attractions, like between the different strands of DNA, those are like attractions. And so those are, we call them like hydrogen bonds, but really they're not really bonds, they're just strong attractions. But the covalent bonds we're forming, like between the actual letters, like linking them together into these chains. This is these are covalent bonds, and these are strong bonds, and these involve the sharing of electrons. So if you have something that is electronegative, like an oxygen, and it binds to something else in a covalent bond, and so they're sharing their electrons. So although we can draw these electrons in like these shells, really these electrons are just like whizzing about in a cloud. And so we can think of there being like this cloud of electron density. When you have a molecule like oxygen that's really electronegative and it binds to another atom, the oxygen is going to pull tighter on the shared electrons. This is going to make the oxygen partly negative, and it's going to make the other thing, so it's going to make the hydrogen in this case partly positive. Yeah, so like with water, the oxygen is partly negative because it's pulling this electron density, and the hydrogens are partly positive because they're having the electrons pulled from them. When you have a situation where you have something that's really electronegative, it can end up, so it's really hoggy, so it's going to take a bunch of electrons, but then it might end up in a situation where it has like more electrons than it can comfortably handle because it only has a fixed number of protons. And so if it ends up with like more electrons that it can handle, then it might want to look for some help, so in sharing that electron burden. So it's like the dog walker has too many dogs and now it wants some help. So it needs to find another nucleus, so some more protons to help out. So we call these nucleophilic because they're looking for some positivity, and positivity is found in the nucleus. And so a great example that you see a lot of a nucleophile in biochemistry is this O-. So if you look back at the DNA leg, you can see that you have that OH group, and if it deprotonates, so it loses that hydrogen, then you have an O-, and it can act as a really good nucleophile. And then you need something that's an electrophile, so a nucleophile is perfect match as an electrophile because an electrophile wants electrons, and the nucleophile wants a nucleus, the electrophile wants electrons, and so they're a match made in biochemical heaven. The electrophile we're going to see in this reaction is actually going to be the phosphorus in this phosphate groups. So although these phosphate groups are negative overall, the phosphorus actually has a positive and is electrophilic because the oxygens are pulling the electrons away from it. So just like we saw with the water where the hydrogens got partly positive, here we see that the phosphoruses get partly positive and are going to be electrophilic. So we have a nucleophile, we have in that OH, we'll have a nucleophile with the O-, and then the phosphate will have our electrophile. But if we look back at the DNA legs, we see that it's in this OH form. Well, typically in this OH form. And so one of these magnesiums is actually going to help activate the end of the chain. Basically, it's going to lower the pKa of this OH group, make it more acidic. And so we talked about pKa's in the context of like proteins yesterday. And so that's basically a measure of acidity. And so the lower the pKa, the more acidic something is. And the more acidic something is, so the more likely it is to be deprotonated. So being an acid and acting as an acid means you give up a proton, you deprotonate. And so if something's more acidic, it's more likely to be deprotonated. It depends on the pH and everything. Check out yesterday's post for more. But so you have this nucleophile thanks to this magnesium. You have a strong nucleophile, you have a strong electrophile, and so you can have this oxygen attack, and this is going to form another bond. And then what you get is you get this, when you have the oxygen attack, now there's way too many things attached to this phosphorus. And so this is going to leave as our leaving group, and it makes a good leaving group. So if this is like resonance stabilized and stuff, and so you can have this be a good leaving group, and then this can break even further to give you even more energy. If you want to see like the molecular details and stuff, I found this cool article that actually shows you like this. They solved the structure, like the crystal structure of a bunch of these like intermediate steps, and so you can see that you have these two magnesium ions that are being held together by these glutamate and aspartate. So there's acidic residues we talked about, so they're helping hold the magnesium. And then you can see that this is the DTTP in this case. So this is the incoming nucleotide. And this is the chain that it's getting added to. So this is your 3' OH. This magnesium is helping lower the pKa of this OH group so that it can get deprotonated and act as a nucleophile. Then you see you have this bond forming. And so this bond is forming and this bond is breaking. So remember with the SN2, you have one bond forming and the other bond breaking. And so this is happening. You have these magnesium ions helping stabilize this intermediate. So now you have this bond formed and this bond broken. And now you can see that you actually have this breakdown of the pyrophosphate into the orthophosphate. And so this is going to be helping provide the energy to drive the reaction and ensure its irreversibility. And I also think that it's helping, the energy from the release is helping with the translocation. So moving the, basically shifting the DNA in the pocket of the polymerase so that a new entropy, so a new DNA letter can come in. So yeah, so check out that article if you want to learn more. And see, you can see that you could have this reaction happen because you already have basically the money attached to this incoming base. But what happens when you have a broken chain or a gap chain and you want to join them together? So maybe this comes into play when we're talking about, this comes into play both in our bodies and in the lab. And so in our bodies, basically, when we're doing our DNA copying, so our replication, you have two strands of DNA and they're copied, the DNA polymerase, so the enzyme that we just talked about, it can only go in one direction. So it can only go like 5' to 3', but you have these two strands of DNA. And so one of them is moving in the right direction. I mean, they're both moving 5' to 3', but because the strands are anti-parallel, one of the strands is going to be moving in the direction of like the growing replication fork. So if you think about a zipper unzipping, one of them is moving like with the zipper and the other has to copy the other strand. And so because the strand is like it's getting unzipped behind it, it keeps having to go back and add stuff in. And so you get these DNA synthesized like discontinuously in these short pieces, and then they have to be added, joined together by a ligase. And so don't worry about all these details. Check out the Okazaki fragment post if you want to learn more. But basically, you need a way to stitch these together. And similarly, in the lab, sometimes like when you're doing restriction cloning, so when you use like sequence-specific DNA cutters called restriction enzymes, and to cut the DNA in specific places and then stitch the pieces together. To stitch them together, you need to use the DNA ligase. And so there are different uses for DNA ligase, but how do they work? So they have to work in a different way because your incoming letter only has a monophosphate. I mean, once again, this should have another negative charge here, sorry. But it only has a monophosphate, and we need to have a triphosphate because we need to have some sort of, it only has a monophosphate, but so there's nothing to kick off and has served as that leaving group. And so remember with this SN2 reaction, you have something added, then your leaving group leaves because it's a substitution reaction. So you're substituting this leaving group for this incoming group. And so we need a leaving group. And here we don't have a leaving group to help pay the cost. So we need to add on a leaving group, and we can do this by using ATP. And so when we do this, what we're going to end up with is we're going to have the release of pyrophosphate like we had before, but also AMP, so adenosine monophosphate. And so how this is going to happen is in a couple steps. And so first you're going to activate the ligase. And so this is going to happen through a lysine in the active site. And so lysine is another one of those amino acids we talked about. What's really great about lysine is it has these this amino group that this nitrogen has a lone pair of electrons and it can act as a nucleophile. And so it can do kind of like what we saw before with the DNA letter attaching to another DNA letter, except here you're having the lysine. This the nitrogen, instead of the oxygen on the DNA, like the 3' OH, here you're having the lysine nitrogen is going to attack this phosphorus. And you are going, this arrow should be starting on the nitrogen. It's going to be attacking this phosphorus, and this is going to get pushed out as this pyrophosphate group, which can then get hydrolyzed to release even more energy. And you're left with this ligase AMP intermediate. So now this is happening in your ligase enzyme site. So now you have this energetic molecule stuck onto your in the enzyme active site. What can happen next is that you are going to activate the DNA. So you're going to transfer the AMP from the ligase. So this energy is stuck onto the enzyme, and now we want to get it stuck onto the end of the chain that's getting added. So we're trying to make this incoming, like this end of the chain, we need to make it have the energy it needs to be favorable for joining to this. So you take this, you want to transfer this energetic molecule, so this AMP part, to the incoming or to the free end of this chain. And so you can do this by having this oxygen attached to here and kick off this, so free the lysine back to how it was. And now you have this attached in this DNA adenylate form. And now you have your chain is still broken, but you have energy to use because now you have something good to kick off. And this ligase is reset, but that's not all it has to do, because now we need to kick off the AMP. So we have the DNA adenylate. So this is the DNA adenylate. So you have this group, and then you have this this broken DNA, and you want to join them together. And so now you have a reaction similarly to have what you had before, but now your leaving group is this AMP, not the PPI, so not the pyrophosphate. So when you do this, you have the attack here, but now you're going to kick off this AMP, and now you have your reaction sealed. So there's one other note. So if your DNA, sometimes like a DNA can break the wrong way, or not the wrong way, but if you have DNA break so that don't have, so if you have the DNA break so that you have a free 3' OH and a free 3' OH and a free 5' phosphate, now you can have the ligase reaction just like we talked about. But if you break it so that you have like a free 3' monophosphate and a free 5' OH, in this case, another enzyme called polynucleotide kinase phosphatase can help add a phosphate group to this 3' OH to make it able to join back together. And so sometimes that'll come into play in the lab as well.